Fountain of Youth. Humans have been searching for an eternal fountain of youth for many centuries with no luck. The fountain of youth is said to cure all illnesses and restore one's youth. But does it really exist? Probably not. Ponce de Leon, in the year 1513, along with his crew, discovered Florida in search of the fountain of youth. However, subsequent research showed that Ponce de Leon and his crew were not entirely motivated by the fountain of youth, and no documents, including a letter written by him, have been found that even mention such a fountain.
In 1938, Herman Muller and Barbara McClintock observed the ends of chromosomes and discovered that the purpose of telomeres is to prevent the chromosomes from attaching to each other.
In 1938, Herman J. Muller used Drosophila melanogaster flies, which were exposed to X-rays at the Edinburgh Animal Genetics Institute. He had observed that a protective cap at the ends of the irradiated chromosomes did not present deletions or inversions. Also, he created the name telomere from the Greek terms telos, which means end, and meres, which means part. 2 years after, Barbara McClintock, who was dedicated to study corn genetics, described how the parting of the chromosomes resulted in adhesion and fusion of their ends. She demonstrated that regardless of this damage, the ends could be restored thanks to the acquisition of a new telomere.
A famous biologist of the University of Yale, initiated in 1978 their work with tetrahymena thermophila phylated protozoa that, unlike other eukaryotic organisms, has an addition of a micronucleus with normal chromosomes. A micronucleus in which fragmented chromosomes are found in multiple small segments of DNA, having the same coding gene for ribosomal RNA. It was then possible to determine the sequence of the extra chromosomal DNA in whose ends they appeared multiple repetitions of the hexanucleotide CCCCAA, those that also were found in the micronucleus. Nevertheless, the finding brought with him doubts. It was perhaps a rare property of an extra chromosomal gene pertaining to a ciliated organism that, in addition, did not form part of the main eukaryota evolutive line. Or it was perhaps an authentic telomeric sequence.
End replication problem. During replication, the ends of the chromosome for the lagging strand cannot be fully copied. This is known as the end replication problem. In this example, the parent DNA strands are blue, newly synthesized DNA strands are green, and RNA primers are red. Parental DNA on the top is replicated. DNA polymerase and ligase will replace all the RNA primers with DNA except the RNA primer at the 5' ends of each newly synthesized blue strand. This means that each newly synthesized DNA strand is shorter at the 5' end than the equivalent strand in the parental DNA.
In 1961, Hayflick and Moorhead challenged the concept of immortality by conducting experiments that discovered fibroblast cultures derived from human skin divided 50 times. After this, they stopped and underwent synthesis. It was also established that the total number of divisions since birth was significant, and not the total number of divisions in culture, since older cells divided less times than younger cells. In 1984, the enzyme telomerase was discovered by Carol Greider, who worked in Blackburn's lab. The political aspect of telomerase research is that after it becomes successful, it is possible that there will be a line drawn between poor and rich individuals. This is because wealthy families are already thriving, and immortality will add to their superiority. Large amounts of money is being put into telomerase research. However, no new discoveries have been made recently.
This area of genetics is being researched very thoroughly, as it will be tremendously beneficial to society and the economy if the key to maintaining telomeres could be determined. The overall purpose of researching telomeres and telomerase is to possibly extend the lifespan of all human beings and curing diseases that are correlated with old age. Extending the lifespan of humans would help society, as people would live longer and thus keep their jobs. Also, medical costs would decrease, as less people would be hospitalized for diseases such as cirrhosis of the liver, leukemia and blood cancers, pulmonary fibrosis, and intestinal disorders. Also, less people would be inclined to retire, more people would maintain their jobs, and as a result, would help the economy.
So where are we now? In 2010, mice with tissue atrophy, stem cell depletion, organ failure, and impaired tissue injury responses were used to scientifically support that telomeres can extend telomeres of chromosomes. The results displayed extension of telomeres, reduced DNA damage, reversed neurodegeneration, and improved the function of the testes, spleen, and intestines by reactivating telomeres in these mice. Thus, telomere reactivation may have the potential for treating age-related diseases in humans. To see that telomerase has been successful in defected mice, it opens many doors in the science field as there is a lot to learn about DNA.